my name is Joe DeWeese. I am Professor of Biochemistry here at Freed-Hardeman University. Really excited about our summer research program and the amazing students that we have. We've got several fun things we're working on this summer. We're working on Topoisomerase 2, which is an amazing enzyme that untangles your DNA. And we got a lot of experiments to run on some new versions of that, some mutations we made to that enzyme to study how it works. We're also working on extracting and sequencing DNA from different sources like fossils and bones. And we got a whole team of informatics students helping us process that data and figure out what's in it and what does it mean and try to understand it better. So I'm super excited to see these students grow and to see what they get to do this summer as part of this program. It's going to be a lot of fun. So.
3, 2, 1. Day 2 of the summer research program at Freed-Hardeman University. I'll be walking you through what we do here, working on ancient DNA. Our friends back here are working on topo research, and it's very exciting stuff, and I can't wait to walk you through everything that we do back here. We are also working on a base-calling model, so I'll also take you through the bioinformatics team that is here on campus. Stay tuned.
I'm Ethan.
I'm Dylan Darty.
What's up? I'm part of the summer research program. This summer, I'm actually continuing some of the research I did last summer. Basically, I spent all of last summer making these generations of Topoisomerase 2 beta. It's one of the enzymes that we study. Right now, we're comparing the CTD, also known as the C-terminus domain. It's intrinsically disordered. Basically, it can be a big mess sometimes. We're doing different things to it. We're adding different mutations, adding DNA to see how that influences it. And Clay's helping me write a paper about it.
Working on similar things to him. My research is focused a little bit more on Topo2A, which is a related enzyme to Topo2B, which he's been focusing on. We're going to be running some experiments using fluorescence anisotropy, which is basically just a way to kind of measure how much the enzyme binds to DNA. And we've created some mutants to see how mutations in certain areas will affect that. So yeah, it's fun. We should do it.
100 mils and pipe it off. Control stock, so we're freezing down the yeast that has our topo mutants in it, so that if we need it later, or if we have it, kind of like a storage. But once you've got your pellet, which we put in the centrifuge, you will pipe it off 500 microliters or half a mil of the liquid. Don't touch the pellet because that's what we need to stay there. Then you just want to put it off into a beaker, and then we'll take bleach from it later. You can use the same pipette for both of the. We're gonna take 500 microliters of glycerol stock. Yeah, there is, it keeps the DNA and the topo in here from like separating and breaking. So it basically keeps it alive while it's frozen, while it's freezing down.
About research because it's so interdisciplinary, and so like earlier today we were using microscopes, and then we're doing a lot of like chemistry stuff with the centrifuge. So just the whole mixing everything that we've learned in different classes into one thing.
Did online schooling for like the last half of my high school career, and so the only lab experience that I had was like freshman biology. I took chemistry. I did not have any lab experience because I was online for me. And so all my lab experience has been this year, and so I'm playing catch up a lot. Like some of the other ones here, they do have research experience, and so I'm playing catch up. But he, like Dr. Dewey's and everyone else, is very patient, so I can go and ask him like the stupidest questions imaginable, and he doesn't. He's not like.
Right.
But yeah.
Hey guys, it is day 3 of ancient DNA research at Freed-Hardeman University, and we are already making mistakes. So
we have EDTA buffer that we're making here, and yesterday we made a mistake of adding too much EDTA to the solution, and so today we're gonna dump it out and make new solution. And I'm saying this because just to show you that mistakes are gonna happen. Like Dr. Dewey says that count all your costs. And so that just shows that you're gonna make mistakes in the lab, but it's important to embrace it and move on from that and grow from it. What we're doing differently now is that we're adding sodium pellets. And just to give you an idea of what we're doing with the EDTA, we're making solution, the buffer that will help extract the DNA. Right here we have sodium hydroxide pellets. So this is basically used to increase not necessarily increase, but to neutralize the pH. So we want the pH to get to 8. And so these gradually increase the pH of the EDTA on 7.23, and that is perfect. So we don't have to add any of this in.
What am I doing?
Mm-hmm.
We're working on Phoenix. We're just training on them first, though, so that we get
The thing I worry the most about is not so
Right now
much
we're
like
doing
the
drug
gel as a whole,
positions.
but the
And then
wells,
we'll
like if it has
start on the master
solidified
mix,
between
the linear no.
Do y'all have a base call model that does this? No, we don't. Are you planning to make and the main thing we're doing here is we're just making sure that the solution just mixed up doesn't have anything in it that we don't want to get in with our process. Okay, so it's bacteria, viruses, all that kind of stuff. We want to eliminate whatever we can. So, all right, where's our solution?
This right here.
Oh, it's in here. Yeah. All right, here, you pour it. 'Cause you worked hard at this. That's it. So, like this one.
Like the whole thing?
Yeah.
Hey guys, it is day 4 of our research. It's been a very slow week, but it is worth the wait. We continue our research on topo, and we are still soaking some bones. Super excited to see some of the DNA that will be sequenced. We also have our bioinformatics guys still working on some base calling model. So stay with us, be patient, and get to see the end product at the end of the summer.
Okay, perfect. We got 6.
Progress. Got some progress.
Yeah.
Got some topo fluorescently labeled, so we're ready to do some stuff.
Let's go. Thank you. Right here, we have a DIY moment. Sometimes you're gonna have to think outside the box to figure out your solution to the problem. This is nothing complicated, but this is just to show you that not everything is perfect in a lab setting. You know, you're gonna have to think outside the box within the boundaries of the ethics of science. And so we just had a weird DIY moment. So we are getting some DNA out of these bones that we have gotten in some of these tubes right here. So we got tubes right here, and these are supposed to soak, and we're supposed to get DNA out of these so that we can sequence them. We're gonna test and see if it works. So fingers crossed. Let's do it.
Bottle with no put the black lids on, and they've got little metal things at the bottom. That's what'll sit down in the bottom of the bottle. So then it gets sucked in.
I'm doing something. What are you doing?
What we've learned about research is documentation. So documentation helps our experiments to be trusted because they would be repeatable, and other scientists can look into our work and be able to observe at the processes that we've observed. And we look at different things like PCR condition. contamination controls, and other valid mistakes that we might have made throughout the experiments. And so one thing that we take pride in is honesty and integrity throughout the course of our research.
Oh, I love
Okay, how do y'all feel about having movie night?
Right now is we have put our topo yeast cells in an extraction buffer, and we're now freezing them down with liquid nitrogen in this like Yeti liquid cryo cup, and we're mashing it down to kill the yeast and get the topo out of it. So once we crush this down to a powder, it will go back in one of these tubes. So here it looks like kind of, if you look inside, it looks more like a powder. But that's kind of what we're aiming for, is that powder. And later on, we'll start running it through the system in the other room and purifying it into this topo.
This is like DNA, and it's made to what they're interested in.
I study Parkinson's disease is characterized by two major hallmarks. So
Hey, we've had a great week. It's been super busy and we're super blessed. We've hit some roadblocks along the way, but students have been creative and flexible, and we've worked through. And that's what happens in the summer programs. Our goal is to figure things out, and nothing is linear, right? Everything has hiccups. We're actually in the last step here of our protein purification, waiting for these drops of protein to come off of this column here. So this is one of the things we've been working on this week. But we've done several things. We've harvested some new mutants that we'll prep over the next couple weeks. We've fluorescently labeled topo. We've done some work toward base calling and things like this for our DNA sequencing. We got some sequencing samples soaking, and we're also training on some of the experiments we need to study this topo. So a lot of good things going, a lot of parts and pieces coming together. Next week we'll do some more of this, some more purification. We'll run some more topo assays, probably fluorescently labeled some more topo and test it out, and hopefully get closer to, on the sequencing side, sequencing some new samples, but then also doing some base call training where we can teach the sequencer and the algorithm what to look for to see ancient DNA. So we're really excited about that. So a lot of cool opportunities coming.